Chapter 2, Basic Principles and Concepts 1, Parasite Systematics, Ecology and Evolution. Parasite Ecology, Part 4. Parasites have to get from one host to the next, okay? As we saw at the end of the Part 3, with those different, generic life cycles, somehow parasite has to get from the definitive host to an intermediate, to another intermediate, maybe to a vector, so they can get to the final, the next definitive host that will allow for propagation of the species. Over the millions of years of adaptations, over the millions of years of evolution, parasites have evolved some tricks. One of the tricks is overproduction. Parasites are going to overproduce offspring. I mean, look at us humans. We're kind of wimpy when it comes down to it. Two, male, female, come together, may produce here in the States, what is it, 2.4 now? Unless you're a Duggar or something like that. Europe is even lower than that. Africa still, a couple more than that. But still, it's, compared to the animal kingdom, it's still quite low. Parasites, because they cannot, there's no, it's not, probability is low. Good Lord. 1, 2, 3. When it comes to parasites, there is the probability that their offspring will not be able to find, will not be able to get to the next intermediate. There will not be the spread from intermediate 1 to intermediate 2 to definitive host. So there is this overproduction of offspring, overproduction of eggs, oocytes, so that the chances are at least 1 or 2, maybe 10 will get to the next host. Of those, once 1 or 2 may get to the next host. And then of those, and so on and so forth. Increased chances of the continuation of that lineage, of that species, is going to be down to numbers. So we find that it's inversely proportional. The number of offspring compared to the probability of success. Success being going from intermediate to the next intermediate to a definitive host. When those chances are at the lowest, success chances are at the lowest, that probability is low, offspring production is going to be sky high. So that there's the chance of propagation. There's the chance of that life cycle being completed. When the probability of success is actually quite high, well, there's no need to spend all that time and resources making lots and lots of eggs, lots and lots of oocytes. So fewer. So we see fewer need to be made. Fewer are going to be wasted, hoping that at some point in time, the next generation will be able to find a host. When it comes down to the adaptations that are going to increase the chances of success, we see that asexual reproduction is a good one. Okay? In the larval stages, you're going to see asexual reproduction. One oocyte hatches, gets a larvae. That juvenile larvae finds one snail. Given time, that one larvae that got into that one snail, 1 becomes 2, 2 becomes 4, 4 becomes 8, and next thing you know, you've got a couple hundred that are going to be leaving that snail, going looking for the next host. We also have hermaphroditism. Okay, many of the parasites are going to be both true male and female. In higher order organisms, if you have a hermaphrodite, say like, mammals, you end up with something that, genetic anomaly allowed for a hermaphrodite, a true hermaphrodite, they're going to be sterile. But in parasites, hermaphrodites are sexually mature and able to fertilize and be fertilized. Some of these parasites can self-fertilize, think similar to a plant and its self-pollination. But most of them that are hermaphrodites still need to. They get together. Parasite A gives up, sperm, fertilizes parasite B. Parasite B at the same point in time will be fertilizing, donating sperm to A. Why would they do that? Well, the chances of parasite A, parasite B meeting up in somebody's large intestine, small. So the chances that they need to find somebody else, means that their chances of being able to propagate are low. And as I said, some parasites have increased their chances of procreating by allowing for self-fertilization. We also see what is referred to as Shizuogany, okay, where during asexual reproduction, instead of one going through mitosis and giving you two, you end up with one giving you 4, 5, 6. One divides, has a nuclear division for, 2, 3, 5 times before there's any type of cytokinesis, before there's any type of dividing of the parasite. So the nucleus divides many, many times, and then you'll resolve this into individual cells, individual organisms. You can end up with also what is referred to as budding. We see this with what are referred to as cysticerci, or cysticercus of Tania. These are going to be the small larval stages that can penetrate into tissues in the intermediate hosts. They will go through rounds of division via budding. Think of it making a mini-me that's going to break off. The mini-me will now grow, increase in size to become a fully formed cysticercus itself. And those two will then bud off others, and it'll keep going and going. So that what started off as one larvae, one cysticercus that got into the tissues of that intermediate, given enough time, can go through divisions via budding, and you end up with a few dozen different parasites now, all from the same original, that are now moved throughout the muscles, moved throughout the tissues of the intermediate host. During Chapter 1, discussed hydatid cysts. Iconococcus granulosus is the one that can cause these hydatid cysts. One cysticercus goes on a walkabout inside the host, gets lost, winds up someplace. One buds off two. Those two will start budding off 4, 5, a dozen each. And it keeps going and going, and you end up with this pocket that's going to be filled with what looks like liquid, with what looks like little grains of sand, except those little grains of sand, each one is a different cysticercus. And then you can also get what's referred to as polyembryony. One larvae goes in, and instead of true divisions, that one larvae will develop differently. Think of it, it goes off on a tangent, and that tangent is becoming a sporocyst. It will physically change its shape, change its metabolism, so it now starts to host different larvae that it will develop with inside it. It will burst at some point in time, releasing these new spores, these new myristidia within the intermediate host. To also increase the chances of reproduction and transmission, as we stated in the last lecture, you're going to have an overproduction of eggs. Tapeworms. Each of the segments, if you look at a tapeworm, each of those segments, as we're going to see in a later chapter, are referred to it as a proglottid. Each proglottid has both ovary and testicular tissue. They can self-fertilize. So that way they start producing and popping out through a pore the fertilized eggs. Okay, oocytes. Each of those eggs, each of the oocytes has the potential to develop into a new tapeworm, but not in that host. They have to be passed out, go through some larval stages, taking up intermediates before a new definitive host. Well, the chances of one of those fertilized eggs not only completing its development to become a larvae, and then those larvae finding an intermediate host, are quite low. So we end up with an overproduction. Turns out tapeworms can produce thousands of eggs per proglottid. And you think each tapeworm, that can be anywhere from, a couple of feet up to 15 feet in length. And each of those little proglottids, about 1/4 of an inch, 1/2 centimeter in length, maybe even smaller, depending upon the species. Each of those in that 2 or 3 meter length of tapeworm, each of those is going to be producing thousands of eggs a day. So in essence, one tapeworm can produce hundreds of thousands of eggs per day, every day, for months on end. Overproduction. To increase the chances, one's going to make it. At least one will be able to complete the life cycle. So you think about it, hundreds of thousands a day, how many millions of eggs are going to be shed by somebody who's sick with a tapeworm? And tapeworms aren't an acute, I quickly get it and it quickly passes, goes away type of parasitic infection. This is a chronic. This is something you can have for months, years on end. So that way you're producing millions of eggs per year for years. Other organisms, other parasites like, say, the trematode. Trematodes are going to, certain trematodes such as Dichloridium, are going to alter their intermediate hosts in some way. Alter the intermediate host in a way that will increase the chances that intermediate host gets eaten by the definitive host. Dichloridium. That parasite, the larvae, will go up into the tentacles or the eye stalk of a snail. Because Dichloridium, the parasite itself is banded, has colorations. Once it gets up into that relatively clear tissues of the eye stalk, it will be there and it will start pulsing. The parasitologists think that pulsing that changes will catch the eye of a bird. By catching the eye of the bird increases the chances that that snail will now be eaten by a bird, the definitive host of Dichloridium. Another great example is, of these adaptations. These ones here we're talking about that are the behavioral. This is the best one. Okay, this one is one of those that is the true. I think falls under the zombie classification. A parasite that turns an ant into a zombie. I mean, it's still kind of living, but it's not completely lost all control. Dichloridium Dendriticum. Okay, it's a trematode. Its intermediate host is an ant. What it does, it gets in the ant, gets into the head, changes the brain chemistry of the ant. Turns out many ants during the day they're out. At night they go back to their nest, or vice versa. They're nocturnal, hidden during the day, they're out, night they're hidden. Well, what Dendriticum does to these ants is it changes its brain chemistry in such a way that the ant no longer wants to go and hide in the nest. It wants to go and climb up on top of the tip of blades of grass and then just sit there. Why? Because grass gets eaten. Grass gets eaten by the herbivore of choice, sheep. Turns out common sheep are the definitive host for Dichloridium Dendriticum. Dendriticum. Dendriticum changes the behavior of the ant, increases its chance taking. Okay, normally many insects and things are going to be very low on taking chances. Okay, if it hasn't been done by another ant, another insect, they're not going to do it. They always follow the leader. Here, it's going to increase, it's going to lose fight or flight, or the whatever ants have for fight or flight, and it's going to increase its chance taking, risk taking. It's no longer risk adverse. It's going to go and it's going to climb up to the top of bladed grass, increasing its chances of being. As in all things, we've been talking about macro, micro. We're going to see that the epidemiology, the spread of parasites, also can be thought of as a macro versus micro. Macro epidemiology is looking at populations as a whole. Micro, the individual. What's the epidemiology within an individual or a small subset, a few individuals, a small unit? Whereas macro is looking at broad scopes, looking at the much bigger picture. As I mentioned before, we're going to, when we're talking epidemiology and transmission, we have to take into account that how things are going to be passed, how things are going to be transmitted. It's going to be impacted by host age, sex, socioeconomics, diet. Somebody who is halal is not going to be exposed to Taenia saginata. Taenia saginata being the pork tapeworm. Okay, if they're halal, they're not eating pork products. Has to be taken into consideration. Also, ecological conditions. Okay, if some of the larval stages require fresh water. I'm in a desert. I live in Nevada. There's no standing water here. There's no fresh water here. Things like that. Not something you're not going to have to worry about schistosomiasis, which requires standing, almost stagnant water like you're going to get in rice paddies. Epidemiology is spreading into what they call theoretical parasitology, mathematical modeling. Okay, we're taking what we have looked at and what we've counted, and we like to say, oh, we've counted everybody. No, we haven't counted, but we've taken subsections of a population that we've sampled, and now we need to be able to figure out ways to apply that to the whole. Theoretical parasitology, mathematical modeling. Here, from this subset, we can now calculate what the population as a whole is right now, or was last year when we took the samples and we counted. And we can look at model historical. And if we have historical, we have current, we can predict future. What's transmission? What's the outbreaks for the future? What is going to be the spread of certain parasites? based on mathematical modeling.